Hello, everybody. Welcome to the Brain Space Initiative. I'm Vince Calhoun, and it's my pleasure to introduce. Now, Amy, please correct me. Is it Amy Kusieski?
Yeah, very
All
good.
right. Ooh,
Yeah.
okay. So Amy is an associate professor of math in the radiology department at Weill Cornell Medicine. And she's also got an appointment in the computational biology department at Cornell University. She did her PhD at Case Western, and she's been working for over a decade in just trying to understand how the human brain works. She's interested in working towards improved diagnoses, treating of neurological disease and injury. So she's been focused in the area of mathematical modeling, looking at different neuroimaging techniques, one of my favorite topics. And she's looking with particular interest in things like lesion symptom mapping, non-invasive brain stimulation, and pharmacological intervention. And so she's going to be talking about some of that today in her talk called Towards Causality in the Brain from Lesions to Psychedelics. And so without further ado, Amy, I'll let you take it away. And thanks for joining us.
Awesome. Thank you for having me. It's a pleasure to be here. And I will just edit. I actually changed my title this morning because I forgot about another aspect of my work that can be considered causal, and that's
Oh, yeah.
image presentation in the brain. So I'm going to talk a little bit about that today too.
Great, great.
But I will start with like a very interesting fact that I saw the other day, which is that 90% of the scientists that have ever lived are alive today. So we are in a very large group of really dynamic people. And the bar for becoming a scientist obviously has gotten low over the years. Some argue could get better, but things are getting better. So, even
Wow.
60 years ago, I couldn't, I might not have been able to be a scientist. And so this is a very exciting time that I think we're living in, especially in terms of neuroimaging and publicly available data sets. So when I started in neuroimaging in 2010, it was very difficult to obtain data sets. I had to basically just cold email people and ask them if they could share their data with me. And you might expect that it didn't go so well all the time. And so when the Human Connectome project came out and I understood what they were doing, I was just floored and so excited about the fact that I could have my hands on this large data set that was very broad range of behavioral and cognition and multimodal MRI. So I was super excited about that. And then since then, there's been a whole bunch of different publicly available data sets that have become available, including things in disease and disorder, like the Enigma data set that tries to kind of make these big data sets that are pulled from different sites. And then of course, the things like in development with the ABCD data set and the Child Mind Institute, and of course, ADNI looking at Alzheimer's disease. And then even one of my favorites is this 28andMe data set where they've got this sample over 30 days of a female participant and hormone levels. So we can actually look at this densely sampled data to see how hormone levels may impact the brain. And so just a little kind of schematic of this. Of course, the number of subjects and the hours of fMRI data per subject, just from an economy standpoint, is sort of exponentially negatively, a negative exponential related. So of course, if you have densely sampled data, you can only do so in a smaller number of subjects. Of course, our ideal is up in this right-hand corner. Someday we might get there, but we are definitely taking steps forward in publicly available data sets. And of course, these data sets usually give us multimodal information about brain function and structure and anatomy. And oftentimes in these data sets, we have fMRI, task fMRI, or resting state fMRI, task-free fMRI. Some also have MEG and EEG that measure brain activity over time. And of course, in fMRI, we have all of these limitations that we know of. Physiological noise, motion can be a big problem, especially in people who have some sort of disease or disorder, and then also in developing populations. If any of you have children, you know that it can be very difficult to get them to sit still. And so these are all kind of limitations in the measurements that we're making. Also, there's statistical sort of limitations where you have to be careful about the number of comparisons and multiple corrections that you make sure that you do multiple corrections for these comparisons, because you are measuring over hundreds of thousands of voxels. And of course, measuring brain structure is another aspect to these data sets where you look at anatomy and or white matter connectivity. So looking at different white matter structures in the brain with diffusion MRI and a process called tractography. And here I just have a little video going through our structural connectome where we have pairs of discrete gray matter regions, and we are showing in blue there the streamlines that are connecting those regions. And so using this tractography applied to the diffusion MRI can give us an idea of the structural integrity of the connections between different brain regions. And of course, there are limitations there that we know about, including trouble tracing through crossing and kissing fibers. And then there's been a ton of studies looking at how tractography can often result in many false positives. So there's a lot of work looking at global filtering and different techniques to try and limit these errors and limitations. But currently, there's not really a gold standard. And then, of course, one of the major goals of these publicly available databases are to do brain behavior mapping. And in brain behavior mapping, we have the MRI, we extract their biomarkers, and then we also have the cognitive and physical or behavioral measures from those subjects. And we use things like statistical modeling and machine learning to relate those MRI-based biomarkers and our cognitive and physical behaviors. And in my opinion, there are two kind of major areas that we want to explore in this type of brain behavior mapping. One is in clinical application, where we're trying to create more accurate diagnoses and prognoses, and perhaps even personalized treatment selection, or even develop novel treatments. And then the other is just more of a basic science understanding of brain behavior relationships. And so these large data sets, and I'm sure all of you are aware of the current back and forth in our field about how big our data sets need to be before we can reliably test our hypotheses. But I just want to bring us back to and remind ourselves that you can actually obtain a lot of information from an N of 1, right? So we look historically back and we see that there are these specific cases of N equals 1 in history that have given us lots of information about how the brain works. And so it might be argued even that the concept of brain behavior mapping happened with Phineas Gage, which we all know, like the railroad spike went through his frontal lobes, and he had personality changes that persisted after he recovered. So that was maybe the first idea that there might be a contribution of the frontal lobes to personality and behavior. And then, of course, with Broca's subject LL, where this person could only produce the word tan. And then after that person died, they were able to identify the area of lesion, and that is now called Broca's area. Also, speech comprehension, Wernicke did that in a similar way. And then more recently, things like memory and learning, we learned a lot from Henry Molezan, who was a person who had bilateral hippocampal damage. And then also there's other cases like SM, where they have bilateral amygdala damage and they have no fear and positive affect. And then, of course, things like how vision works, blind sight, things like that. So really, we shouldn't lose sight of the fact that N equals 1 can give us some information about how the brain works, especially in disease populations. And so, of course, looking at patients, advanced MRI can be done in these clinical populations, and they can provide information about how these networks change in disease and possible compensatory effects or reorganization that might drive recovery. However, it is difficult to collect these types of data sets in patient populations because we know that pathology can increase SNR in ways that might obscure underlying connectivity changes or changes in the brain. Patients can't tolerate these long scan times often because they're in an acute stage of anxiety because of their disease or disorder might have just happened, like a traumatic brain injury or stroke. And then, of course, the other side of this is the economy side of it, which is that it does cost money to do these advanced MRI techniques, and it requires a high level of expertise to process. So we wanted to figure out how we could leverage this large number of naturally occurring lesion studies to understand how the brain works and devise more accurate prognoses or more effective therapeutics. So to that end, a while ago now, I came up with this idea of the network modification tool, or the NEMO tool, that allows us to estimate the structural disconnectivity due to a particular lesion just from that lesion mask itself. So without any kind of diffusion MRI or structural connectivity extracted in the patient, we may be able to estimate how much that lesion disrupts the structural connectivity. And it's been recently updated by my lab member, Keith Jameson. So here he has updated it to work on a web app. So you don't actually have to download any software. You can just go and upload your MNI co-registered lesion masks. And what it does is it takes that lesion and it looks at 420 unrelated human connectome project tractography sets of tractography results. And then it sees the streamlines that pass through that lesion and then where those streamlines end and begin. So it can actually give you a voxel level measurement of disconnectivity for each gray matter region, gray matter voxel in the brain. It can also summarize that at a regional level or a global level. So if you're interested, you should definitely check that website out. And recently, there's actually been a lot of work in this area as well with similar types of approaches. And we've applied these, and other groups have used our tool to apply them in different disease and disorder populations. So things like stroke and predicting cross-sectional impairments, as well as future impairments, and looking at traumatic brain injury and MS and kind of mapping those lesions, disconnectivity profiles to their behavioral outcomes. So one of the more recent studies that we've been doing that's in preparation right now is from my student Emily Olafsson. And here we were looking at predicting cognitive outcomes after stroke in a mostly chronic population of stroke subjects. So here we took the auditory working memory scores, these Ways total digit span scores, and we used ridge regression with a type of feature selection on those regional disconnectivity measures from the NEMO tool that we got just based on the person's lesion masks. And we you put that into our ridge regression model to see how well it predicted each of these or that outcome from each of our different modalities. So here we have the structural connectivity that's estimated from the NEMO tool. And then here we have a PCA version of the lesion mask itself, lesion volume, and just the clinical variables. So you can see that using clinical variables and our estimated structural connectivity from the NEMO tool actually gives us the best explained variance. The other thing that's interesting about this is we can look at the health transformed regression coefficients to identify a variable importance. And so here we actually see that if you have damage to left subcortical frontal connections, then you will have a worse overall outcome. So there's also another tool in the literature that's used by Mike Fox and Aaron Bowes and their groups looking at how the functional connectivity may be related to the lesion location. So this is actually the functional kind of mirror image of our network modification tool, where you take a person's lesion mask, you co-register it into a common space, and then you look at how that lesion's time series is correlated with the rest of the brain. And what you get out is then a voxel level volume of how correlated that particular voxel is with the lesion's time series. And so this has been applied in many, many different studies. They've really done a great job with this. Things like even free will or prosopagnosia, Holmes tremor, and they found some really interesting results. One recent one was actually in addiction and cessation of tobacco use after stroke. So that was kind of something that was really interesting in the literature recently. I think it was covered in the New York Times. And so this estimated structural connectivity, you might say, well, why isn't it better to just do the advanced imaging and MRI in subjects? And so my previous statements about how it might not be feasible logistically or money-wise. But there also might be some information that's obtained using direct fMRI in the subject, so about how maybe recovery works. And so this paper actually compared head-to-head the explained variance in the outcomes based on estimated structural connectivity and estimated functional connectivity, and then actual functional connectivity that was observed in the patients themselves. And they argued that the estimated functional disconnectivity didn't do as well as the estimated structural connectivity or the real functional connectivity. But there have been a ton of papers since then, kind of going back and forth. So there's a lot of back and forth in the literature right now, and it's an interesting time because there's a lot of papers coming out in rapid fire succession, saying that one is better than the other, and maybe not. So this is still an open question, an area of active research. Okay, so up until now, I've talked about how lesions may impact structural and functional connectivity and how we might actually estimate those values just based on the lesion location itself. But of course, the relationship between structure and function can inform how we have an impairment or how we recover after injury. And so there are many different ways that people have used to relate structural and functional measures, including things like linked neural mass models, statistical and machine learning approaches, graph theoretical models, and even network control theory. So the structural connectome, of course, is the backbone of activity propagation within the brain. So you can imagine that if there's a damage to that structural connection, that it may also alter the flow of functional connectivity. There was this really interesting simulation study using neural mass models that were linked by structural connectivity measures drawn from diffusion MRI, and they simulated the effects of lost structural connectivity on functional connectivity. And there has been some more recent studies in stroke, specifically looking at voxel level identification of the effect of the disrupted streamline. So here you can see these two yellow voxels are connected with this blue white matter connection, and you see that they do have a high correlation. Then they showed that if stroke damaged the streamline, that you had a loss in that functional connection. So of course, this is not a surprising result, but it's really interesting to see this analysis at a voxelized level. So there are a lot of studies that looked at the structure-function relationships in disease and recovery, including neurological and psychiatric disorders, both. So two studies that I'd like to highlight here that use the virtual brain framework. That's the neural mass models that simulate functional activity at each region, and then they're linked by the structural connectome. And those two studies showed a correlation between the model parameters that they fit using the patient data and cognition in Alzheimer's and motor impairment after stroke. TBI is another example. There was a recent study that showed damage to the structural connectome can actually decrease metastability of the brain's activity patterns, which was then correlated with worse cognitive outcomes. And then, of course, there have been a lot of studies in depression and disorders of consciousness showing that stronger structure-function coupling was related to worse outcomes in both depression and disorders of consciousness. So first, I'd like to focus on one of my results and modeling approaches, which is the network diffusion model, which is a graph theoretical technique. And so here, up in the upper right-hand corner, there's a little animation of this type of modeling approach. So network diffusion is not something new. This is something that has been applied in graph theory for a while. And it basically states that if you have some activation or some substance at a certain point in space, that it will diffuse along its neighbors. And that diffusion will be proportional to the amount of connectivity between those two nodes and the difference in that level of concentration or activation between those two regions. And that's exactly captured with this differential equation, which applied to the whole brain actually has this really nice closed form solution involving the Laplacian of the structural connectivity matrix. And so this is the exponential of the Laplacian. And so here I just have a little animation where we have in the upper left-hand corner the observed functional connectivity for a patient, upper right-hand corner the observed structural connectivity for a patient, and then down here you're going to see the network diffusion model process on the structural connectivity. And then here you're going to see the correlation between predicted and true observed functional connectivity. So you can see as the network diffusion happens, we do get a bump in that correlation. And of course, it's not perfect, but it does allow us to kind of measure the alignment between structural and functional connectivity. And the alignment, we kind of have in the past summarized the alignment by finding the value in the network diffusion time that gives us the maximal correlation between predicted and true. And what we do is we call this network diffusion model propagation time. So the smaller this is, the more you might think that structure and function are aligned. And the larger it is, the less you might think they're aligned, because you do have to let that process happen longer and longer to get a good correlation with the true observed functional connectivity. So what we did was we actually found that this model propagation time, which we kind of interpret as capturing the alignment between structure and function, was positively related to better outcomes after disorders of consciousness, after coma in patients with disorders of consciousness. So we looked at all kinds of graph theoretical measures, and really the only one that was significant after corrections was this model propagation time. And we actually solidified our results by doing a simulation where we virtually removed structural connections in the brain and their functional connections, their corresponding functional connections. And that was what we said was an injured brain. And then what we did was we replaced the functional connections and said that that was a recovered brain. So we were assuming that if you lost a structural connection, you weren't going to get it back. But if you're recovered, that you might have replaced that functional connectivity, functional connection. And so what we did was as we removed more and more structural connections in this framework, we see that in our simulated recovery case, the model propagation time does increase, similar to what we found with our actual data. In a more recent study, we applied the same kind of approach in mild TBI that we had longitudinal data for. So here our hypothesis was that over time, if a person had more increases in their network diffusion model propagation, that they would have better recovery. And that's exactly what we found. So we did find in this set of kind of relatively small, but it was a longitudinal data set, that increases in model propagation time, as well as increases in functional connectivity degree and decreases in functional connectivity modularity were associated with better recovery. Okay, so let me switch gears a little bit and talk about stroke. So stroke is a little bit different than traumatic brain injury because there's this very localized acute area of damage, and you can usually identify it based on an anatomical scan. And so this really cool paper that showed that white matter structural disconnections can actually explain network dysfunction. And of course, this is not a surprising thing, but it's really interesting to see them quantify this relationship. So what they did was they took the structural areas of damage and they said, okay, so how the areas that have a structural disconnection, do they also have functional connectivity that's different from control populations? And they did find that 39% of these functional disruptions could be predicted by structural disconnectivity. And interestingly, they found that when they looked at not just the direct connections that were lost in the structural connectivity due to lesions, but the indirect ones by using network diffusion, they actually found that that was even more related to functional connectivity damage. So there does seem to be a direct connection disruption that in structural connections that affects the direct connection as well as the indirect ones. So that brings me to my next work that I'd like to discuss, which is using longitudinal sort of densely sampled data after stroke to look at functional connectome reorganization. Because this can allow us to understand how the brain might recover lost function so that we can devise strategies to support those mechanisms. So we know from animal studies that adjacent cortex can take over the function of damaged neighbors, but thus far, this type of reorganization has not really been shown at a whole brain level in humans. So in this study, we wanted to investigate using this novel approach called graph matching and to see if it could possibly uncover these types of mechanisms. So we took 23 subjects that had first-time pontine stroke. Then they had resting state fMRI and motor assessments at 1 week, 2 weeks, 1 month, 3 months, and 6 months after stroke. And then we applied graph matching to their extracted functional connectomes at subsequent time points to identify regions whose functional connectivity profiles may have been taken over by other regions at the follow-up time point. So here you have a little schematic of it, where at 1 week post-stroke, we have this region I that's denoted in green, and it has this particular type of functional connectivity to the rest of the brain. And then we look at the next time point, and we actually see that a different region, region J, has a very similar functional connectivity profile to region I, and region I has changed its functional connectivity profile. So using graph matching, we can actually identify this type of swapping of functional connectivity profiles to get an assessment of how much functional reorganization is happening over time. So our first hypothesis was that gray matter regions that were more impacted by the stroke via their disconnectivity profiles may also have more remapping over time. And so we indeed see that this is true. We saw that there was a positive correlation between the baseline amount of white matter disconnectivity and the amount of functional remaps at each pair of subsequent time points. So those regions that had more structural disconnectivity due to the lesion then went on to have more functional remapping per this graph matching technique. Our second hypothesis was that more remapping may be indicative of more severe impairment, and possibly that that might also relate to better recovery of motor function after time. So we did see that this was true. We did find a significant correlation between baseline impairments and early remapping in the 1 to 2 weeks time points after stroke. And we saw that early remapping was related to the amount of recovery in the chronic stages. And also using a linear mixed effects model, we put all the time points together to show that the amount of remapping between subsequent time points was related to the change in the motor scores and in such that more functional reorganization over time was related to better improvements in their motor scores. So again, this was done by my student, Emily Olafson. Okay, so another type of approach of relating structure and function that I want to touch on is using machine learning. So there was this really interesting paper in 2020 that was using an artificial neural network to predict functional connectivity from structural connectivity. And so we had this tool, the network modification tool, that could take a lesion and give us an estimated structural connectivity. So the insight we had was that perhaps we could use this neural network to then create an estimated functional connectivity from a lesion mask. And so we did this. This is work by my postdoc, Jaren Toslu. And we did this in a set of 100 people with MS. So we had their stroke lesions defined in a semi-automated way. We put it through the NEMO tool to get their estimated structural connectivity. Then we put it through this artificial neural network to get estimated functional connectivity. And in that same data set, we had the actual subjects, diffusion MRI and resting state fMRI, to actually get their observed functional and structural connectivity. Then what we did was we applied a classification model to classify the MS subjects into disability or not disability. And surprisingly, we found that estimated functional connectivity and regional estimated structural and functional connectivity did better than our observed functional connectivity and structural connectivity in classifying patients with MS into those disability categories. And then we also have, and that was published earlier in 2021. And then we have some more recent work looking at second set of subjects from 171 patients with MS who only had lesion masks and cognitive scores. And we were actually able to achieve a state-of-the-art explained variance in predicting cognitive scores from the estimated structural and functional connectivity using just the clinical MRI. So that was all using this SAR-WAR approach to predicting functional connectivity from structural connectivity. But my lab member, Keith Jameson, decided to come up with a different framework. And this is actually a really interesting approach that uses a joint autoencoder that maps the structural and functional connectivity flavors. So there's a bunch of different flavors. And he maps them into a shared latent space. And then you can actually get back out any one of these arms. And so we call it the crack encoder, which is like a brilliant name for it. Keith came up with that. So I think the key insight here is this push-pull loss function that exists in latent space. And so in the latent space, what we did was we added to the cost function a penalty that suggests that the same subject's multiple flavors should be mapping to a very similar point, and that similar point should be very different from the rest of the subjects. And so here we trained it on Human Connectome Project data. And what we did to assess the accuracy was we looked at top one identifiability. So if you take a person's structural connectivity in the test set and you predict their functional connectivity, how many times does that functional connectivity correlate maximally with its own functional connectivity? And then of course you can also look at average rank identifiability. Okay, so here these plots are showing on the left here the top one accuracy, and on the right the average rank identifiability. And these are all the different flavors of functional connectivity in the top three rows, and then the bottom two are the different probabilistic and deterministic tractography approaches for structural connectivity. And you can see that the top one accuracy for predicting structural connectivity from functional connectivity is about 41%, and predicting functional connectivity from structural connectivity, the maximum we get is about 30%. And this might not seem that impressive, but I wanted to emphasize that the current state-of-the-art modeling gives us about 2.5% top one accuracy. So to go from 2.5 to 40 is quite a feat. So I'm very proud of Keith's work here. And then of course the average rank identifiability is also very high at about 90% for both of these, and the current state-of-the-art is at 60%. Okay. So the last thing I want to talk about in terms of structure-function relationships is network control theory and quantification of brain state dynamics. And so here the idea is to take our entire time series, and instead of collapsing it down into a single matrix that represents the correlation between time series, we actually analyze the spatial activity of the brain throughout time. And so here in all of these works that I'm going to discuss, we basically pool everybody's time series together that are extracted on a regional basis. And we put everybody together and we apply k-means clustering to identify commonly recurring states of brain activity. So then for every subject, you get now a time series of states that tells you the state that person's brain was at any point in time. And of course, if you have this time series of brain states, you can then calculate things like fractional occupancy, or the amount of time that's spent in a specific state. Things like dwell time, how long you spend in a state once you get to that state. And also appearance rate, so the number of times a state appears per minute. And then you can calculate more fancy things like transition probability. So how likely is it for a subject to transition from, say, state 1 to state 2? And so this work, the first work that I'm going to talk about is by my student Emily Olafsson. It's in press now at ComBio. And what she did there was she put the 23 subjects with pontine stroke and the controls as well, and found 4 optimal states that seem to be dominated by frontoparietal activity and motor activity. And she analyzed the differences in the fractional occupancy of this frontal parietal state, which is the one she was more interested in, because there is some evidence that the frontal parietal activity may help stroke subjects to recover from motor impairments. And so we actually see that stroke subjects throughout all of the time points after stroke spent less time relatively in this frontal parietal state than their controls. However, interestingly, more time spent in this frontal parietal state for those stroke subjects was correlated with better motor recovery, but only in patients with dominant hemisphere CST damage. So the thinking there was that when you have dominant hand CST damage, that means that the functional reorganization might mean that you're going to take over areas close to the motor cortex and use those for performing motor tasks. And in the dominant hand, we know that there's a larger spatial representation of motor function in that hemisphere, in that dominant hand hemisphere. And so it may be then easier, more beneficial for those people with dominant hand CSTs to spend more time in the frontal parietal network that may underlie recovery of motor function. So that was all looking at brain states individually and looking at fractional occupancy. But we can also then, once we have these brain states, use network control theory to assess the amount of energy demand needed to transition between different brain states. And so this was a work that was presented by Danny Bassett's lab in 2020 in this really beautiful paper in Comms Bio, where they analyzed the amount of transition energy needed to transition between states based on the structural connectome and network diffusion on that structural connectome. And they actually found that there was a relationship between age and whole brain control energy, wherein it seemed to get easier over age to transition into the default mode network. So this is kind of an interesting approach using brain state transition energy and looking at kind of things like demographics, age, and other things. So we decided to apply this in our own data. I see that I'm getting a little bit short on time, so I'm going to try to skip this. If anybody's interested in the exact details of how to do transition energy, please contact me. So the first study, this was in about 100 MS patients with 19 controls. This is done by my postdoc, Jaron. And after identifying these 6 states using k-means clustering, we calculated transition energy between all pairs of states, and then we used logistic regression to predict class from the transition energy matrices. And we actually found that people with MS without disability had lower overall transition energy than controls, but people with MS with disability had higher transition energy than controls. And we interpreted this as perhaps a functional compensation mechanism that drove TE lower, the transition energy lower in the people with MS without disability that then exhausts as MS progresses, eventually leading to the MS group having higher transition energy than controls. We also interestingly found a negative relationship between global entropy, so the entropy of the brain activity time series, and energy, indicating that a person that had a higher energetic demand to transition between brain states also had lower overall entropy, which is sort of unsurprising. And then in the people with MS, we found that there was those that had larger lesion volumes also had decreased global entropy. So I'll get to the psychedelics now. Understanding and being able to measure these mechanisms of psychedelics is really important if we're going to use them in a therapeutic context. And you can see here that there have been a dramatic increase in the therapeutic studies of psychedelics. One relatively consistent finding in the brain activity analysis of psychedelics is that there's an increase in entropy under the effects of psychedelics. And it seems to be related at a regional level to both the white matter structural connectivity and the 5-HT2A serotonin receptor density, which is where the psychedelics have the most robust agonistic effects. So one of these models of action in the brain is known as the relaxed belief under psychedelics, or the rebus hypothesis. And it was created kind of to explain these findings. It views the brain as a prediction engine, whereby the perception and belief that you have about your environment are shaped by both prior knowledge of your environment and incoming sensory information. Within rebus, the psychedelics are postulated to decrease the prior beliefs, which then enables the bottom-up information flow. So it's theorized that the observed increase in entropy and brain activity is actually reflective of reduced energetic demands for the brain to navigate its dynamic landscape. So that's really what we were most interested in. One little tidbit I'd like to talk about, because my brain is currently under this the effect of this drug, is that caffeine also increases the entropy of the brain. And that was a study done in 2018. So our hypothesis is that this work, by the way, is all done by my brilliant PhD student, Parker Singleton. So our hypothesis was that perhaps psychedelics observed effects of increased entropy in the brain is arising from a flattening of the brain's energetic landscape. So in this scenario, you might imagine your brain activity is represented by the coordinates of a hiker within a landscape. And the shape of that landscape will determine how much energy is required to move from one point to another. So if you have a very steep landscape, you're going to require a lot of energy. And if you have a flatter landscape, then you're going to require less energy. And it might result in increased entropy. So in this approach, we use network control theory to kind of assess the landscape. So quickly, just the LSD experimental design was 20 healthy volunteers who underwent MRI scanning sessions at least 14 days apart. In one day, they were given intravenous placebo, and the other, they received intravenous LSD. And then they had a mock scanning session, and then followed by a true scanning session. And then they had resting state fMRI. So in the end, we had 15 subjects because one was excluded for anxiety and then 4 for excessive motion. So in this study, we had 15 subjects. We also had a separate study that was in psilocybin, and this was in 9 volunteers that were scanned over 2 days with placebo and 1 with psilocybin. And they had 5 minutes of resting state. So here, again, these are the states that we found by applying data-driven k-means clustering to the brain activity time series. And we saw that after LSD, the subjects' brains spent more time in a somatomotor sort of bottom-up state than they did in a frontal parietal sort of top-down state. We also found that across both LSD and psilocybin, that the transition energy required to go between every pair of states decreased in all cases for both LSD and psilocybin. So interestingly, this is a really nice visualization of what's happening in the brain. So here we have the time series, and the y-axis is the amount of energy required to transition between subsequent TRs. And each line in here is a subject. And you can see that the placebo infusion does not change the amount of energy required to transition between subsequent time points. But after psilocybin, we definitely see a large shift downward in the amount of energy required to transition between subsequent time points. And so then we also showed across the population that the amount of energy reduction in these transition energies was correlated with more dynamic and entropic brain activity. So this was across individuals. We found those that had larger decreases in energy also had larger increases in entropy. So by what mechanism we wondered, do transition energies decrease? So we do know that the psilocybin and LSD act as agonists on the 5-HT2A receptor. And we do have a mapping of that from PET imaging from a totally different set of subjects. And in network control theory, there is this ability to assign a level of control that's different across the different regions in the brain. And so in our hypothesis, we wanted to see what happens if we assign a level of control to each region in the brain that's corresponding to the amount of 5-HT2A receptors. And so this is the original result where we showed that the transition energy decreases with psychedelics. Then we looked at the placebo centroids only and compared the transition energy of the uniformly assigned control weights and the 5-HT2A weighted control weights. And we saw that there was a dramatic decrease still in the amount of energy demand to go between pairs of states. And then we compared it also not just to the uniformly weighted, but a spin permutation on the 5-HT2A receptor weights, and showed that there was still a decrease in the amount of energetic demand. We then compared it to other serotonin receptors, and we found that the 5-HT2A does seem to have a spatial distribution that is somehow optimal to flattening a landscape. So this work should appear shortly in Nature Communications. So there's also been a lot of work in looking at MDMA. So I know that I'm up against time. Do I have a few minutes to cover some things?
Sure, please go ahead.
Okay, and please, if people want to interrupt me, I'm totally fine with that.
Sure.
Okay, so a lot of studies have looked at the effect of MDMA for treatment in PTSD. So this is something that's been happening a lot more lately. So we looked at this data that we had from a small trial run by MAPS, looking at the effect of MDMA therapy on neurosignatures in the brain. So they had baseline and follow-up after MDMA therapy. They had baseline and follow-up resting state and task fMRI. The task fMRI design was a script-driven autobiographical memory recall where they had a neutral script where they were just reciting what they did in their morning routine. And then they had a script that was describing the traumatic event for which they were seeking therapy. So they actually showed that the CAP score, which is a measure of PTSD severity significantly decreased at follow-up in response to therapy. And we also showed that there was bilateral hippocampus and left amygdala increases in functional connectivity after therapy as well. And then we wanted to look at the task fMRI design. So we identified these 4 regions of interest from previous task fMRI studies in PTSD. And then we calculated the voxel-wise t-statistics for these regions for the trauma versus neutral. And we saw that at baseline, there was definitely higher activity during the trauma script versus the neutral script. There were a few areas of significance. And then after therapy, the trauma versus neutral didn't show any differences. While there were no differences in the baseline and follow-up contrast maps, there did seem to be an overall trend in the decreased contrast at follow-up. So this was kind of a small N study. And we did show also that larger decreases in the PTSD severity across subjects was related to larger decreases in right amygdala bilateral precuneus functional connectivity during the neutral listing task. So there does seem to be some evidence that the amygdala is playing a role in the reduction of PTSD severity in response to MDMA therapy. Okay, and so this is the last thing I'll talk about. So I added this on at the end because I was thinking to myself that, of course, when we present images to a person's brain, that's also causal mapping. And so, of course, encoding and decoding in the brain is a hot area of research, especially with the large explosion of research in artificial neural networks. And so encoding is just when you take an image and then you look at the brain's response and you try to predict the brain's response to this image. And then decoding obviously is the other way around, where you have a brain activity pattern and you want to reconstruct the image that elicited that brain activity pattern. And then, of course, in machine learning, there's a lot of work in generative adversarial networks. I'm sure some of you on Twitter have been looking at the DALL-E 2, where you can type in text and it gives you back an image. So there's really a lot of very recent and very interesting work in generative images. There's also been some interesting work in monkeys trying to manipulate neurons with generated images. And so here, this was a really cool paper by Carlos Ponce. And here they picked a single neuron that they could record from, and they tried to create these generate images that would stimulate that neuron to have the highest firing rate that they could. And as you can see, as the evolution goes on for the different units, you get like a different image, but they do seem to be consistently perhaps monkey faces, especially the first one does look like a monkey face. So there's this really interesting work going on now to manipulate neuronal activity based on images and using generative networks to do that. So here we looked at the natural scenes data set. This is no longer a preprint. This is in Nature Neuroscience. So sorry about that. But it's a densely sampled data set of 8 subjects who had 30 to 40 MRI scans over a year's time. And they were shown between 20 and 30,000 images. And so this is this beautiful, huge data set that we can then use to create encoding models to predict brain activity from images. And that's just what we did. And then we also added it to a generative network. And this is what we call neurogen. So this was just in Neuroimage earlier this year. And here what we wanted to do was create a framework with which we could use the encoding model information and a desired brain activity pattern, and then create an image that would give us that desired brain activity pattern. So it's sort of creating an image that gives you a specific pattern of brain activity. And so the first validation we did, I want to mention this is by my really brilliant PhD student, Zijin Gu. And so what we did first was we did a single region activation maximization. And here we have FFA, the space region, body region, parahippocampal place area, word regions, and V1V, of course, that responds to textures. And you can see, it's kind of hard to see because it's a little small, but the natural images at the top were the ones that gave us the biggest response in the actual data for the specific region. And then the second one is the natural images that gave us the best response in the encoding model, gave us the highest predicted activity for the FFA in the encoding model. And then these are the neurogen results that are the synthetic images that achieve the maximal activity in that particular region. So you can see things basically, they do align with what is expected. So FFA does have person, EBA does have bodies, parahippocampal place area does create place images, etc. And so what we did then is not just single images, but we could single regions maximization and minimization, but we could then do pairs of regions or even 3 regions together. So here we just have an example of 2 regions, and the orange is the observed activity for the natural images. or actually I think it's the predicted activity for the natural images, and the blue is the predicted activity for the synthetic images. So you can see that the blue are always further from the origin than the orange. So we do seem like we may be identifying something in the synthetic images that may lead to a maximal activation in that particular region. One of the things we did see, which was super interesting when we did the synthetic images for the face areas, was we saw a large representation of dog images. And so at first we thought it's just a byproduct of the big GAN, because big GAN has an overrepresentation of dogs in the set of images and categories that it represents. But then we said, well, let's actually test to see if that's true. So we actually calculated for each individual the T statistic between the activity in that particular face area when they saw an image of a dog versus an image of a human. And we actually found that the number of dog images in the top 10 synthetic images was correlated with the T statistic. So those people who had regions that responded more to dogs versus humans then also had more dog images in their top 10 synthetic images. Okay. So to summarize, we use a wide variety of methods to investigate causality in the brain, including lesions and pathologies, stimulation, non-invasive images even, and then looking at the effect of longitudinal treatment or natural recovery, as well as pharmacological studies to understand how you can manipulate brain activity. And of course, measuring or estimating these connectomes and their relationships and these biomarkers may allow us to better understand what actually causes impairment and what may drive recovery. And then that will help us clinically to improve our management of these diseases. And so I want to just make sure I advertise my 3 students are going to be graduating next year. It's very sad, but they are looking for postdocs. So if you're interested, please reach out to me or them. And with that, I'd like to thank my funding and my lab.
Thank you, Amy. Wonderful talk. I really appreciate that.
Yeah.
So I just want to mention that because there was a question that the video is being recorded and will be uploaded in YouTube and Bilibili. And if you have a question that you would like to ask yourself, let me know to unmute you. So I have one question relating to structural remapping in stroke. They are asking if that was a mild stroke with or without physical therapy, and asking if you can provide some information about post-stroke treatment.
Okay. So if you're talking about the pontine stroke lesions.
I think so, yeah.
Yeah, so
Like
all of them had a pontine stroke lesion. They were varying sizes, but they were all pontine strokes, and they all had motor impairments. And most of them did recover over time. There were still a few that didn't recover to the full amount. And of course, there's always this ceiling effect when we're looking at behavior in stroke, because you can recover quite rapidly. But Emily actually came up with this really brilliant way to assess the ceiling effect, and found that what we were doing was predicting above and beyond just the ceiling effect in recovery. So I hope that answers the question.
I think so, yeah. So another question is about that functional remapping. I think it's about the time that you were trying to explain, like one region replaced the other one. I think they were asking if you can explain more about that.
Okay, let me see if I can go back. So this is an approach called graph matching, and it's independent actually of brain imaging generally. It's just it's a graph theoretical approach, and this approach basically tries to sort of reorder so if you have 2 matrices. In this case, we have 2 matrices from our neuroimaging study, but you can just think of them as 2 matrices and two that represent graphs. Then there's a lot of interest in trying to identify a mapping between the networks, right? So you want to figure out if this network's region is the same as another network's other region. And so there's a lot of work in trying to create a mapping from one network to another. And so that is basically what we did here, which is that we tried to identify regions that had functional connectivity profiles or rows in that functional connectivity matrix that were more similar to another region's row in the subsequent time point. And so you actually just swap the rows and columns and identify the permutation that gives you the maximal correlation between the two the remapped functional connectivity and the other functional connectivity. So it's a way of reorganizing the functional connectivity matrix by shifting the rows and columns that aligns it maximally.
Thank you. And we already shared the paper, so if you are interested, you can look at the paper for more information for sure. So I have a question for myself, if it's okay. So I was wondering if you ever look at the possibility that one region does not have the same functionality over time. We call it spatial dynamic. So usually like what you have is you assume these region having this same activity over the time, and then either using CAP or other approaches, you try to find like represent across time. But I'm wondering if there is a possibility that the region have a different functionality over the time. And
yeah, so this is a really really good question, and it was something that we were wondering as well. So in this paper, we actually had healthy controls that had longitudinal scans. And we were able to perform this graph matching and identify that regions that have lower SNR within the functional MRI also do more remapping, right? So it's not unsurprising because there's lower SNR, so you're going to have more uncertainty in the mapping of their functional connectivity profiles. But so what we wanted to do in our study was identify the amount of remapping that was only due to the stroke. So what we did was we calculated the remapping and functional connectivity remapping in healthy controls, and then basically just took out all of those connections in the structure in the stroke subjects. So this particular study was not interested in looking at the functional connectivity remapping in the subjects, but it was interested in just the stroke part. So there could be some longitudinal changes in the functional connectivity remapping, and we actually have a preprint. A manuscript in preparation right now where we're doing inter-subject functional connectivity. And we also looked at test-retest functional connectivity in the HCP subjects. And essentially what we're interested in is sort of not the longitudinal aspect of this. So we tried to just remove the effect of any kind of test-retest remapping. But there really could be, over time. I'm not sure what the time frame would be to like observe a real remapping of the functional connectome in a subject, but
right. So and also I think we can look at it within session. Like for example, if you look at the network, how a network formalizes over time, or how these nodes within a session can change, like something like a dynamic, the same concept.
Yeah. I actually had this idea too. So kind of looking at sliding window dynamic functional connectivity and then applying the graph matching, that'd be really interesting too.
That's my research area. That's what I was curious about.
Yeah. I think it would be really interesting, actually. We should talk more about that. Yeah.
Sure, definitely. Thank you. And there is another question, if you don't mind. They're asking if you can go through the energy transition measure. I'm not sure if you have a time to briefly explain that.
I do have like 1 or 2 minutes, so let me go through it. So the interesting thing of this, so this is in a paper. So there's a couple different papers that use this now, but essentially what you do is you assume that the brain is a linear system where activity X at time t flows passively through the network via network diffusion on the structural connectome A, and actively through the effect of external inputs U that drive the system. So you have internal network diffusion, and then you have external control inputs. And so then what you can do is this is not something that was created independently. Like the network control theory is something that has been developed outside of this, right? So looking at this controllability Gramian, you can see that this exponential A, this is network diffusion solution, right? And so actually what this transition energy is representing is the amount of energy needed to inject into the system to complete the transition from the naturally diffused initial state on the structural connectome and the state in question, the final state that you want to find. So it's really the amount of control energy that you need to inject into the system via U to get from your naturally diffused initial point and the final point. So I hope that helps. And if you need more detail, you can definitely look at those original papers.
Thank you so much, Amy. I really appreciate your time. If you have more questions, feel free to continue, ask your question, share them on the Slack. And thank you for your time. I really enjoyed it.
Yeah, it was a pleasure. Thank you so much.
Thank you. Have a wonderful weekend, everyone. Bye-bye.